Epistasis is the interaction between non-allelic genes at two or more loci, resulting in one gene masking the phenotypic expression of another gene. There is that one gene or allele masking the phenotypic expression of the other genes or alleles in the interaction. That gene or allele masking the effect is referred to as epistatic. When dominant allele A masks the expression of B, A is an epistatic gene of B. A can express itself only in the presence of B allele. Therefore, it is called dominant epistasis. B expresses only when a is present. Therefore, in a 9:3 to 3:1 ratio, both 9 and 3 are expressing the A gene. The ratio is now 12:3:1. This is known as dominant epistasis. This connects back to Chapter 5, where dominant epistasis is seen in the interaction of two loci that determine fruit color in summer squash, which is of three colors: yellow, white, or green. And that is shown right now. When a homozygous plant that produces white squash is crossed with a homozygous plant that produces green squash, and the F1 plants are crossed with each other, the following results are shown. In the F2, 12 out of 16 of the plants produce white squash, and 1 out of 4 of the plants produce colored squash. This outcome is the familiar 3:1 ratio produced by a cross between the two heterozygotes, and it suggests that the dominant allele in one loci inhibits the production of pigment, resulting in a white progeny. This also connects back to Chapter 6 and pedigrees. As in dominant epistasis, the gene at one loci masks the expression of another gene at another loci, and it inhibits its expressivity. It can connect to the pedigree in such a way that it can show up in different frequencies. A pedigree chart is simply a diagram that could show the occurrence and appearance of phenotypes of a particular gene or organisms, and phenotypes of its ancestors from one generation to the next. They are used to analyze the pattern of inheritance of a particular trait throughout a family. This could include mutations, disease, etc. Epistasis and recombination also relate. The competition between epistatic selection and recombination in the outbreeding phenomenon is the focus of the study. In the presence of epistasis, selection, by increasing the frequency of favorable genotypes, establishes correlations between alleles at different loci. Recombination, however, reshuffles alleles and randomizes genotypes, breaking up co-adapted loci. Because the recombination rate between any two loci is largely determined by their physical distance on the chromosome, the effect of genetic interactions depends on gene location. Functionally related genes tend to cluster, suggesting selection on gene order. Chromosomes have different regions of infrequent recombination. This is how it ties back into Chapter 7 and recombination.